thanks, Bob. I hope I'm up to that. I'm not so sure. So presenting something very different from the last talk that you heard, not perhaps directly relevant to your own health and well-being. Also don't have as many cool slides to show because I'm going to get into content very soon. But I wanted to start with a painting. It's a painting I like a lot. My wife hates it. So it's in the Boston Museum of Fine Arts, and it's considered the masterpiece by Paul Gauguin, and this is its title. So where did we come from? What are we? Where are we going? Either visionary or pretentious, depending on what you think. It could be the title of a lot of things. It could be the history of vasculitis treatment and how much it's progressed in the last 40 years. It could even be the history of the Vasculitis Foundation. But in fact, for me, it's how I'm going to try to put together the different types of research that go into vasculitis and how the research that we fund through the Vasculitis Foundation is directly relevant to all of those areas related to vasculitis. So, and this is where it comes in. Where did we come from? What are we? Where are we going? What is vasculitis? What causes it? And then in turn, what are the outcomes? And these are all highly relevant to understanding the diseases and then making treatments better and making quality of life better. And there are a lot of different ways that research impacts on these different things. So for starters, there's the term epidemiology, which comes from the same root as epidemic. That's where the study actually started, figuring out sources of epidemics, which I at least include who, what, where, when, and why. Genetics has become a particularly important one to look at in recent years as the tools to analyze it have become more sophisticated. I put genetics under epidemiology, even though the tools actually come from the laboratory. Environmental influences, everyone would love to be able to study those and figure them out. It's a much harder thing to study, so there isn't as much research there, but everyone knows that there needs to be. In terms of what vasculitis is, it's not as simple as it might sound, and so there are large projects involved in trying to classify these diseases. We have certain names for them, but we probably group them and split them apart in ways that are artificial. The features of different diseases, what percentage of people with a given disease are going to have these problems? Do they accumulate more problems over time, or do they tend to be stable? Those are very important areas of research also. And finally, in outcomes, there are, of course, treatment trials, but there are also clinical outcomes that need to be followed. Do patients have higher incidence of coronary artery disease 10, 15, 20 years later, even if they're doing well? A lot of other things. And then an area that unfortunately has become a very important area of study in the last 15, 20 years or so is called generally health services research. Even if we know how to treat people, can we get them the medications they need? Can we educate them properly? Can we get people to, the payers to pay for it? That sort of thing. And so there are a couple of areas of research that kind of fall between these two, leading between cause and what the actual disease is. And this is where a lot of laboratory research comes in. And laboratory research involves a lot of things, as I say, biological molecules, the cells, study of the tissues themselves, and development of animal models. And then something that I do, but no one actually knows exactly what it is, although they all like it, which is called translational research, which I consider as using the tools of the laboratory to really try to address problems that are as closely related to patient health as they can be. And so biomarkers is one of those, trying to use the tools of the laboratory to develop laboratory tests and really test them to see if they're helpful, using the animal models to try to develop new treatments, that sort of thing. So you have your choice in funding science between funding projects and funding people, and there's, not surprisingly, not really a dichotomy there. But at the top, it's probably just as well that you can't see what's on there, because that's a bunch of logos of pharmaceutical companies. And so, as you can imagine, drug companies tend to fund projects, and that's appropriate. They want to try to answer particular questions that are going to be relevant, and things that, to be honest, are going to make them money later, in addition to helping people's health. The NIH is somewhere in between. The NIH funds proposals on the basis of the merits of the proposal itself, but also the track record of the investigator factors into that. And they also have training programs, which really are a balance between what this person's done, where they are going, and how good the project is. I just bring up in passing an organization called the Howard Hughes Medical Institute really makes a point of saying that they fund people. They do so on the basis of their past track record, but they then say, we're going to give this outstanding person a tremendous amount of money, and they don't have to justify what they're doing every few years. So how does the VF fund things? And I think we're really right in the middle, which is really how things should be, and I'll explain why. The First thing is that the projects that we fund are generally $50,000 for 1 year, $100,000 for 2 years, and that sounds like a lot of money, and it is a lot of money certainly if you're a donor and contributing to it. But if you think about it, that is, if you're funding a person, that's maybe one junior investigator or one postdoctoral fellow, that's the term we use in the laboratory, for a year. Or taking a lab that already has money to support all of its people, one very expensive project from a technical side in terms of purchasing expensive materials, that sort of thing, for a year. And where that has value is what I think it's analogous to venture capital. This is something that is going to allow a research group to do something that's innovative that may then lead to something much bigger, or to keep a junior investigator or get them involved in research in vasculitis. And that's really important. That's what a lot of these grants do. They come from the major labs, but support a younger person doing a good quality project that's going to keep them in the field. Part of why that's of interest is that most of the vasculitis researchers, the people who've been supported by these projects, are also clinical experts in vasculitis. They do both. And so that's of direct benefit to patients, not only in the future as knowledge advances, but also on a day-by-day basis. So I want to go through some of these things one at a time. I'm not going to talk about everything that's been funded over the past, I think it's 13 years since this program has been in place, but some areas where there have been more than one project, some that are brand new and in progress, and some that have been completed. So one area where we've funded a fair number of things is in genetics. And here's some examples. So in the genetics of granulomatosis with polyangiitis, or Wegeners, the first study was by Catherine Siminovich, who is at the University of Toronto, and it was called Identification of Novel Wegeners Granulomatosis Susceptibility Genes. It was clearly before the disease was renamed. 2 publications have come out of that, and it involves screening for what are called common genetic variants as risk factors. So I can't go into genetics in great detail, but we all, throughout the human population, about every 50 or 100 bases in the meaning components or letters in the DNA code, we have some random changes. And some of those are associated with different diseases, and some are not. And there are very fancy ways to screen for those. And Kathy's group came up with several, as did a group in Europe at about the same time. And you get some answers, but you don't get the whole answer. And sometimes you might want to look for rare variants or unique genetic variants that are found in a particular individual. And so Sharon Chung at University of California in San Francisco is in the process of doing a VF-funded study on that, using a technique called exome sequencing, where you actually sequence the individual DNA of the part of the genome that actually encodes for proteins. And so that obviously is a work in progress. One of the main things, the consequences of this program, though, is that these studies have led to additional funding for both Drs. Chung and Siminovich, and have kept them involved in the genetics of vasculitis. So Dr. Chung actually got what's called a Career Development Award, one of those awards from the NIH that helps to sponsor people in addition to their projects. She sees patients with vasculitis at UCSF. Dr. Siminovich is actively involved in collaborations with our group and with others. So these people are very much involved in genetics of vasculitis still, in part as a consequence of these projects. This is just an example of one of the things that was learned from those studies. I'm obviously not going to explain all of this in detail, but the HLA is a region that is involved in pretty much any autoimmune disease, but there's just different types of HLA molecules. And Kathy's group, in collaboration with others, found this particular region here, and conveniently at about the same time, the group from Europe found the same thing. So it's real, it's important. Where that's going to go in terms of understanding the disease and treatment is not clear, but at least that particular component of genetic risk is very well defined. The VF has been very important in funding the first studies of the genetics of Takayasu's arteritis, and this work was led by Amar Swala, I believe initially in Oklahoma and now at University of Michigan. And he led the first large-scale genetic study in Takayasu's using something called the immunochip, which had every possible known genetic variant related to immune disease, and that subsequently led to what is known as a genome-wide association study, where every known common variant is popped onto this chip, and 300,000 or even 1 million of these variants are analyzed at the same time. And both of these studies are now published, and they're really it in the field. They're not necessarily the most exciting findings. You find 3 or 4 things out of the million that you were looking for, but it's absolutely essential to have as background for trying to understand diseases better. And so here's an example. These are called Manhattan plots, where this is everything across the entire genome. These are all of the, I don't know if it was 300,000 on this one or a million, and it's called a Manhattan plot, as you can imagine, because of the buildings that come up. But in autoimmune diseases, I think of them more as a Dubai plot, where there's always a giant building right in the middle that represents the HLA, which I said is different in every autoimmune disease. And all of these things here, if they pop up, I think of are more likely to be the diseases that they're called polymorphisms, but the common changes in the genome that differ from person to person are more likely to be related to different groups of autoimmune diseases in general, and this is kind of the one that tells you which one you're going to get. The total genetic risk known in the different forms of vasculitis is quite small. I want to stress that. We're never going to screen people for it, but it helps us understand the diseases. Is Takayasu's more like rheumatoid arthritis or like lupus? Is there something here? Like, this was a rather large surprise. There's not known to be anything related to autoantibodies in Takayasu's. It's not thought to be an antibody-mediated disease, and this is a polymorphism that is related to how antibodies are dealt with and causing inflammation. So that's interesting. We'll learn something about disease and follow up on it. This is the second study that they did in follow-up of the VF-funded study, where they found a couple of other things to look out here, which including an interesting one in interleukin-6, since this is an inflammatory protein that people are studying the blocking of as treatment for Takayasu's. You notice the other one disappeared. That's just the nature of the immunochip versus the GWAS. It wasn't actually a false positive on the last one. So to get into the laboratory side, I think I can talk about the true basic science research and the translational research at the same time, because they use the same tools, and because no one gets very far from studying human disease in this field. And so the type of laboratory research that's been done, funded in general in vasculitis, involves immunology or the immunopathology. I'm sorry about the long words, but I think, I hope it's not too hard to understand, but how the immune system goes wrong in inflammatory diseases. Then looking at the specific targets of autoimmunity, the best example of this is the ANCA antibodies and what they specifically attack in just 2 diseases. These are the only diseases in which we know what the targets are. The ANCA-associated vasculitis, or GPA and MPA, and cryoglobulinemia, specifically only if it's associated with hepatitis C, not the other ones. So there's a lot more to be learned there. There are animal models, and there's a lot more research being done on samples from patients, whether that's blood or urine or tissues. And I just wanted to mention, so that the study of these kind of things benefits from laboratory-based researchers who are well aware of the technical advances that are being made, the basic knowledge of how the immune system works, for example, how blood vessels work. And because they stay in touch with that when new tools are developed, then they can apply them in vasculitis. And so there is kind of a gray area between basic and translational research in vasculitis, because the people who do it are so close to studying the human disease. So here's some VF-funded studies on the immune cells in GPA and MPA, and this is going to be a mouthful. So first of all, I don't know if I need to read this, but I suppose I could. So analysis of, I thought it was something different, but NKG2, which I actually don't know what it is, on CD8, CD28 negative T cells, etc. This was done by Peter Lamprecht in Germany in collaboration with a larger group. What it comes down to is this is trying to dissect out what part of the immune system or what part of immune signaling mechanisms is going on in disease compared to what we expect in healthy people. And similarly, the P38 MAP kinase isoforms that Jochen Swarina and colleagues looked at in kidney disease and ANCA-associated vasculitis, this is trying to take what specific part of an inflammatory pathway has gone wrong, and in thinking ahead to not just understand the disease, but how we might intervene. And finally, Alan Salama's group in England investigated the role of, they were called then novel Th17 cells. Now they're front and center in autoimmune and inflammatory diseases, and I'll get to that in another disease in a minute, and studying Th17 cells, which are T cells that instead of making, before it was either gamma interferon or interleukin-4, they were called Th1 and Th2 cells. Another group discovered subsequently making something called interleukin-17. Stay tuned. It may be related to certain forms of vasculitis. It may not, but they wanted to look at it in ANCA-associated vasculitis. And the important thing here is laboratory science tends to proceed incrementally. You have to be extremely detailed in your studies, and one thing leads slowly to another, but over time a lot is learned. And all of these studies were published. It was good investment by the VF, and all of the people involved have remained involved in laboratory research and are still considered leaders in the field. And that's really important because it's sometimes hard to follow these tracks for a long period of time. There's also been some VF-funded research on the ANCA antibodies, and so now that you know that there are targets like proteinase 3 and MPO, it'd be nice to know what specific parts of the protein are involved. The reason for that is that ANCA may not all be created equal, and it may be antibodies to particular parts of it that are important. And this study also led, the term was conformational epitopes in ANCA, which part of the protein is bound, and this, of course, was published, and the investigator remains a leader in the field. And this is a new study that we're funding this year, looking at a possible third ANCA antibody. This isn't ready for prime time either. There was a very high profile publication, but it needs to be followed up to see if it's of importance and relevant to vasculitis. The target molecule called LAMP2. So for translational research, Dr. Wand, who, as Dr. Madison had mentioned, a longtime leader in the field in giant cell arteritis, has studied vascular T cells in GCA. I mentioned Th17 cells before. Her lab found a remarkable difference between the behavior of Th17 cells and the Th1 T cells that will lead to, and it's very active area in her research, I think, continuing, and this is an important advance in the field, and I'll show you a picture in a minute. Another study done by Lindsay Lally, who's a junior investigator working with the more senior investigator, Robert Spira, looked at a particular intracellular signaling pathway molecule called Rho kinase. I can't explain all the terminology right here, but again, I'll show you a picture. And this one is aimed at improving the diagnostic quality of temporal artery biopsies. These biopsies are often negative in people we think really have GCA, and we'd like for a better way to find it. And this, importantly, has led to a larger collaboration with a group in Europe. So we want this seed money to not only be effective and be published, but to lead to larger collaborations and studies that might actually lead to changes in how we manage patients. And this is a new one of our current crops of studies that's being funded looking at interleukin-6, which I mentioned before. It's actually a clinical trial in process, industry funded, to try to block IL-6 and see if it helps in GCA. And so this is from Dr. Wyan's paper, and all I want to show is that there were both Th17 and the Th1 cells. She may say more about this today, I'm not sure. But they were remarkably different in their sensitivity to steroids. So these were implanted into animals, and the Th17 cells disappeared when those animals were given steroids. It's not prednisone, but equivalent, but the Th1 cells did not. So what does that mean? That's something that is, again, going to be investigated. Undoubtedly, her lab has investigated, but it's going to be something to keep in mind in trying to understand this disease and its treatment. This was from Lindsay Lally and Robert Spira's group. The patient clearly has giant cell arteritis. This protein here is all over the place, but we wouldn't need that to make the diagnosis here. The interesting thing is that both of these biopsies are read as negative for evidence of giant cell arteritis, but in this one, where it was really concluded on other grounds that the patient had the disease, it actually still stains. That biopsy that looks normal under the light microscope actually has a lot of this inflammatory protein inside its cells, and this one not so much. So maybe this will improve our ability to diagnose the disease, although a lot more work needs to be done. A little bit about classification and disease features. Some of the things that are being funded by the VF. One is, in the past, was a study in Canada to try to gather together a lot of patients with childhood onset vasculitis, and this really has led to, it was described as a pilot project, and it has become something much bigger and something that we hear about every year at our vasculitis meetings about the progress that they have made. This is an ongoing study, international, big thing called DCVAS, to try to revise the criteria. We may end up with new names for different forms of vasculitis, we may not, but other criteria to separate out what means that someone has PAN versus microscopic polyangiitis. Will there be another name? Things like that. This hasn't really been revised at the level of clinical information since the early 1990s. It's been looked at really from the microscopic level. And then finally, Susa Benseler in Canada has really been a pioneer in looking at childhood CNS vasculitis and is really trying to separate out what the different subtypes of this disease are. And so this is a work in progress also. To get to the clinical outcomes, some of the studies, again, this is not comprehensive, that have been funded by the VF included early on work from the Cleveland Clinic that looked at a large group of patients who either got cyclophosphamide for a short period of time, which was just at that time becoming considered the standard of care. Short time cyclophosphamide, then given less aggressive therapy thereafter, and concluded, as had been suggested from some small trials before that these people generally did quite well. Secondly, we've heard reference to this before, study led by Megan Klaus, very important study that was really hard for people to deal with before, that no individual center sees enough young women or young men to look at the reproductive outcomes. You have to bring a lot of people together and do a lot of online research. And as I think you heard, Dr. Klaus is doing ongoing research in this area through a prospective, meaning we're going to get information from women who conceive and find out what happens thereafter. I'll show you a picture there too. One of the studies currently being funded is a longitudinal study of morbidity and mortality in vasculitis patients using a nationwide registry in England. So this is, again, something you're going to hear about from Dr. Merkel and the BPPRN of how do we properly use these large databases. And the interesting thing is that Dr. Jick brings in she's, I think, the first person I mentioned who's actually not a vasculitis researcher, but more of a big data expert. And so it's good for people to be working together who have expertise in different fields. And this is from Dr. Klaus's paper. I'm just highlighting the things I think of greatest interest, that pregnancy loss does seem to be a bit higher, unfortunately, in women who had pregnancies delivered after the diagnosis of vasculitis. I don't know if all of you heard this yesterday, but that's actually the normal, that the rate of miscarriages is between 20 and 30%. Maybe it's a little higher. Maybe the rate of preterm birth is higher. It's not terrible, which is important. It's important for us to be able to tell young women that. But it's also important to gain more information and more details about this for people to have accurate information. So a little bit, health services research is something we're just getting into now, and this is a study in progress on the impact of healthcare utilization and informal caregiving in primary systemic vasculitis. The initial reports of this are very well received in an international meeting. People are interested. What are the costs of vasculitis, both to society, how much does it literally cost to have people hospitalized, and also to individuals in a more less financial way. And so finally, I want to talk about what has come about more recently through the VF of explicitly funding people. And that's through the VF fellowship program, which has evolved a bit over the years, and which I think is a really great thing that we're doing. And so the goal of this was to train the next generation of vasculitis experts to do both clinical and research training at leading centers. And initially there was a VF-only funded fellowship, one a year, Elizabeth Brandt at North Carolina, and then Rennie Ree at University of Pennsylvania were the first two people funded under that program. They're both giving talks here today. Then starting in 2015, this merged with a separate program that was done through the VCRC, and I think this is a great idea. I say this coming from both sides there, to pool the money in order to fund, because the goals were very, very similar. And try to fund usually a couple of people a year over the course of several years. Again, to go to particular leading centers, have those institutions provide a contribution as well, but to really bring people into the field. And we expect that the first recipient of that will be chosen this summer. And also, family of vasculitis patient Jeffrey Fishbein has been particularly important and interested in funding this fellowship. And then finally, I think most of you heard about this before, but we're starting to make an effort to have individual fellowships that honor particular doctors. And Gary Hoffman is a particularly obvious and non-controversial person to be supported there. And we were just able to announce that the fellowship in his name will be given to Dr. Hiro Tamaki to be supported during the year of 2016 to 2017 after he spends a year funded by other means at Cleveland Clinic. So this is someone who's probably going to be to go off and be an expert in vasculitis as well. So I believe that is it. Thank you.